joined by Professor Mehmet Somel from University Biological Science Department. Mehmet is a graduate of the Biological Sciences Department, and then he moved to Germany for his PhD, and he did a PhD at Max Planck Institute for Evolutionary Anthropology in Leipzig, right?
Right.
So then he moved to China, in Shanghai he moved on his research on evolutionary anthropology again. Then he moved for his second postdoc to Berkeley, University of California, Berkeley. Then he moved back to Turkey and he started his lab in Mehmet. And now he's pursuing his research on ancient DNA, and he recently got many prestigious awards, and he published many papers in really prestigious journals, including Nature, PNAS, Nature Neuroscience, and many others. So I'm proud that he's one of my close friends.
I'm proud.
He recently got the ERC Consolidated Grant, and it's one of the first consolidated grants that's awarded for biological science as well. So please join me to welcome Mehmet, and thanks for him being here for us.
Thanks. Thank you very much, Urartu, for organizing this, and thank you for coming. The work I'll be talking about is will be on ancient DNA. Our lab is also involved in some other transcriptomics and genomic analyses, but most of our focus is now in this field. And we're not working alone. We're part of a wider group, which also includes archaeologists and anthropologists. There are people doing the wet lab, and I'm mainly responsible for the computational work. We're also very tightly collaborating with Hacettepe Anthropology. We also have collaboration with people here with Can Alkan. The main focus, although we also do work on domestic species and other natural populations, our main focus is human history and prehistory. So, of course, most questions about human prehistory, about how ancient societies were organized, how people lived, how things changed over time. This is the subject of archaeology and anthropology. So people study material culture, the architecture of ancient settlements, what kind of tools people used, what kind of rituals they had, how they buried their dead. And anthropologists can also extract information from the skeletons, such as isotope data, what kind of diets people had at the time, how diets might have varied among people living at the same time or in different times, between male and female, between different social groups. So there's already, within the last, archaeology as a scientific field, objective collection of data to understand past societies. That's more or less 100 years old. Anthropology, as we know it, is a bit more recent. Of course, there was kind of skull focus and racist research early in 20th century, but modern day anthropology is also dates back to mid 20th century. And now we're trying to add more data, complement this picture using ancient DNA. So ancient DNA its history also goes back to the 80s, in fact. So when Sanger sequencing became available and DNA cloning, people were interested in whether they could get DNA information from extinct species or mummies early in the 80s. And then when PCR became available, there was even stronger hype about getting DNA out of old samples. And there was this big excitement about ancient DNA, and people were reporting DNA sequences from dinosaurs or from dinosaur times, mosquitoes trapped in amber, this kind of stuff. But some critical minds started seeing funny patterns there. So this is a phylogenetic tree of mitochondrial DNA sequence, supposedly from dinosaur bone. And as you see, it falls within the mammalian distribution. So we have rodent here and rabbit and dog. And funnily, the dinosaur appears to be a sister taxon with humans. So obviously this was contamination, so all this thing that inspired the Jurassic Park film, etc. And the stuff published, I think this was published either in Science or Nature, so stuff published in very highly prestigious journals, it turned out to be totally bogus. Some of them were retracted, some of them are actually, they ended up not even being retracted, but people realized that, okay, there's a huge problem here, and with targeted PCR, it is extremely difficult to really make sure that what you're studying is authentic DNA or not. Contamination is always a problem, and if you're studying humans, it's even more of a problem, obviously, because we have human DNA all around. And so a lot of labs later shied away from this field, especially human ancient DNA was a no-go for many groups. If you asked me 10 years ago if I would be studying ancient DNA of humans today, I would have said no way, honestly. And in fact, Mahin Rakkaya from Metric Chemistry, she was talking about prospects of doing human ancient DNA. I was telling her, no, just forget about it, because it's you cannot replicate your results. You cannot really make sure things are authentic. But then in the late 2000s, things started to change, and that was thanks to next generation sequencing, the ability to produce very large amounts of DNA data all in one run, shotgun DNA data. And there it also appeared that we could actually identify authenticity of the bulk of DNA. Not every molecule necessarily, but overall how authentic or not, how indigenous or not the DNA we were sequencing was, it was possible to predict. So I'll explain more of that. So by the end of 2000s, early 2010s, there was more and more revived interest in ancient DNA. And at the time, I was doing totally different stuff. But when I moved back to Metu 5 years ago, there was a small lab of Ince Togan. She had started doing ancient DNA, and I joined in. And basically, she retired. I ended up inheriting the lab, so we continued. So I'll first give an overview of how the workflow goes. Basically, we work with anthropologists, and they collect for us teeth or mainly skull parts. So the temporal cortex has some dense parts which tends to preserve DNA better than other parts. And then we extract DNA just like usual. You prepare libraries, next generation sequencing libraries, do sequencing, and then there's bioinformatic analysis and, of course, statistical analysis of population genetics. The sequencing, we use Illumina, which is perfect for ancient DNA because it already produces, you sequence short reads. Illumina normally has now 150 base cycles, and most of the DNA we are studying is already broken up by time. You know, the bonds break down, so the average molecule we are studying is about 60, 70 bases long. So it's perfect for us. You can get a lot of information in just one go, hundreds of millions of molecules. You sequence, and then, of course, you need to check what there is inside. And we now know that in most cases, after you die, the bacteria living in the soil, perhaps some of the bacteria also inside you, they colonize your tissues. And most of when you later sequence DNA from this tissue, bone or tooth, most of the DNA you obtain is bacterial DNA. Sometimes it's 90%, mostly it's 99%. Sometimes it's totally bacterial DNA. You don't get any human DNA at all. And of course, you might also have some human contamination depending on how the bone was handled in the lab or previously. But there we are lucky because now we know how the DNA molecules undergo changes after death, post-mortem changes. So not only does the DNA molecule break down over time, but also the cytosines tend to lose their amine groups. So losing its amine, cytosine becomes a uracil. If it's methylated cytosine, it becomes a thymine. And when you sequence it, you tend to find, and okay, this also tends to happen more often in the single-stranded, at the breakpoints, where DNA becomes single-stranded. So when you sequence the DNA, where you expect to find C, you tend to observe T's more often than you would expect. And the older the sample, this is data from Leipzig, the older the sample, that C to T transitions seem to appear to accumulate. And so if you put all the data you collected from a sample and ask at the ends of reads where you expect to see how frequently you find T or some other base. In this library, this is data from perhaps summarizing across dozens of millions of reads. In this case, you observe more than 25% of the reads which ended with a C, they had a T substitution. And as you go inside, the frequency decreases. And C to substitutions from C to other bases, you basically don't observe. So if you sequence a library from a sample and you find this type of profile, it suggests there is no endogenous DNA. If you find this type of profile, it suggests that most of the DNA is endogenous. And there are also further ways that you can test authenticity. I can explain if you wish later. So this was, of course, a big relief for the community. Now we can do shotgun sequencing, and if we find human DNA, we can also, or whatever you're studying, whatever organism you're studying, if you find if you find DNA that seems to belong to that organism, you can further test authenticity statistically. And what kind of questions can be used to address with this type of data? Most of the research until now has focused on demographic analyses, because even with one whole genome from one individual, you can actually tell a lot about the population it belonged to. Because, as you know, our genomes are mosaic. If we were only studying the mitochondrial DNA, of course, it only tells you about the maternal lineage. But if you're studying the whole genome, then it's telling you it's giving you information about all the ancestors you basically had. If you were not inbred, then 10 generations ago, you expect to have 1,000 ancestors, so all of them have contributed some amount of DNA to you. And so just studying your genome, we can obtain information about the genetic profile of all these ancestors, and then we can use your DNA to compare with, let's say, other populations, other published data, and get an idea about the demographic history of your ancestors. So you can study migrations, admixture, population size changes in time, and then you can try to connect this with changes in ecology, in historical events, etc. You can also, if you have more individuals within a settlement, you can also study kinship patterns among these. You can try to understand matrilocal versus patrilocal, for instance, traditions, inbreeding traditions, how these change over time. You can, of course, also study natural selection in humans or in other species you're studying, or in microbes that let's say, infect humans. So this is, of course, this usually demands more information, because you need the frequencies most of the time. So if you want to do selection scans, you need large sample sizes, but we're slowly getting there. So people are producing more data, and I'll give you a small example on that. People have also tried to obtain epigenetic information. When I say epigenetics, I'm not sure, is everybody here biologist? Not necessarily. Okay. So in addition to genetic information that is coded in the DNA, organisms also modify DNA, adding methyl bases or modifying the histones DNA is interacting with to open or close chromatin, so that it's a way to regulate, it's involved in regulation of genes. And methylation of DNA is tells us how active or inactive genes that harbor those methylation marks are, about the activity of genes. And the thing is that you might remember, so when methylated cytosines get deaminated, they convert into T. When unmethylated cytosines are deaminated, they convert into U. And you can use enzymes that specifically remove U from the DNA. So the C to T transitions then you would observe in ancient DNA would indicate that those cytosines are methylated. So that's it's very noisy, and you need very high coverage genomes, ancient genomes, to reliably obtain this information, but this is also something people have been working on. We include it. And then you can also study the bacterial components. Most of that, as I mentioned, is soil DNA, soil bacteria that have colonized the bones, but you can also sometimes find pathogenic bacteria in the tooth especially, or for instance, we have cases of leprosy where you find on the bones the pathogenic marks, and you can extract DNA from the bacteria or whatever pathogen we're talking about. But it's also not super standardized yet. People are working on it. So what kind of questions would this information help us address? I already mentioned demographic analysis is one major thing. And for instance, we could try to understand the interaction between humans and other extinct human relatives like Neanderthals. We already knew that humans evolved in Africa, anatomically modern humans evolved in Africa. We first see their fossils about 200,000, 250,000 years ago, so fossils that you could not distinguish from us. And then some of these groups, they move out of Africa, and in the Middle East and in Europe, West Asia, they encounter these Neanderthals who are, well, pretty close to us, but somewhat different. And there is some time of overlap. They even use sometimes the same caves, but how they exactly interacted, we didn't know. And archaeologists were discussing for a long time whether there was any gene flow between the two species. They were not very distinct, so it was imaginable that they might have, interbred. But this was not really possible to say just studying the fossils. But in 2010, the first Neanderthal, relatively high quality Neanderthal genome was published, and there people used a number of clever tests using whole genome data to see whether there might have been gene flow from Neanderthals to human populations that had moved out of Africa. So imagine these are modern day African populations who have never gone out of Africa, and these are modern day Eurasians whose ancestors had gone out of Africa and had overlapped with Neanderthals for some time. And let's say this is the Neanderthal genome. If there had been no gene flow from Neanderthals to this group, so we can only imagine gene flow from this group to this group, but not to ancestors of modern day Africans. If there had been no gene flow, so then we would not expect Neanderthal genome to be particularly more similar to Eurasians than to Africans. It should be equally distant genetically to both groups. But if there had been gene flow, then we would expect Neanderthals to be somewhat more similar to Eurasians than to Africans. And indeed, so this is the phylogenetic tree. People have predicted both based on the fossil record and the genetic data. Indeed, when you compare non-Africans, these are modern humans, non-Africans, modern day genomes to the Neanderthal genomes, we are a bit more similar, assuming here everybody is Eurasian. You are somewhat more similar to Neanderthals than modern day Africans. A couple of percent Neanderthal DNA is still residing in Eurasian genomes. It's predicted that about 1 to 3% of our ancestors were Neanderthal. Although the species went extinct about 30,000 years ago, still about half of their genome in different parts in all of us is still surviving. And you can, of course, do further stuff. So with Ömer Gökçemen, who's in Buffalo now, we were checking what, among Eurasians, how much variation there is in Neanderthal ancestry. And actually, although the admixture probably happened in the Middle East, modern-day Middle Easterners tend to have somewhat lower percentage of Neanderthal DNA compared to Europeans and Asians. And would you guess why this could be the case? So Middle Easterners tend to have a bit less Neanderthal DNA, and that seems to be mainly because there has been later gene flow from Africa to the Middle East, and that has diluted the Neanderthal ancestry in the Middle East, whereas Europe and East Asia, they have retained their Neanderthal alleles more efficiently. Okay, but as I mentioned, our main well, I didn't mention this. Our main interest in the lab is the population genetics of this region, so the demographic history of this region, and particularly the Neolithic transition. So Neolithic is the new culture that arose after some human populations gave up mobile hunter-gathering, and some of them eventually turned sedentary, so they started living in the same location for a long time, and some of those groups eventually turned to be farmers and animal herders. And this happened in multiple parts of the world independently, in the Americas, in East Asia, but the first event we know from the archaeological record that this transition happened, was in the Middle East, including central Anatolia, Levant, Zagros region, North Mesopotamia. And later, starting from here, this culture spread to other regions. So in Europe, it was delivered as a package. It was not independently invented, but it was farmers. It was farming culture. People in Europe eventually adopted. I have already a slide here. So this was the Fertile Crescent, and the farming. Although today nearly all populations, except for a few exceptional cases, are doing farming, are food producing instead of collecting their food from the outside mainly. This culture only was invented a couple of times in a couple of places. And this process, although, how to say, it's today the basis of all our civilization, and we have so complex societies based thanks to food production. It was not a very simple and a very rapid process. It took over 5,000 years after the Holocene, so after after the ice ages, when the climatic conditions became better and warmer. People in this region, they first started to reside in the same area and exploit their local resources more efficiently. And some of these groups eventually started cultivating things like wheat or barley, and also manage sheep populations or local goat populations. And thus we had a totally new culture. And just to give you an idea about what these people looked like, as I mentioned, until that point, everybody was hunter-gatherer. All human societies we know of were hunter-gatherers, were collecting their food from outside and living in only temporary residences. So these guys, this is a village near Boncuklu, near Konya. The name of the place, the Höyük, is Boncuklu. And this is a village of something like 50 to 100 individuals, pretty small, and they were building these small mud brick houses, pretty small, like not larger than this table, in fact, where you could host only a couple of people. They were fully sedentary, and they seem to have little social differentiation. In fact, most hunter-gatherer groups we know of from modern times or in the past, they are pretty egalitarian, so there's not so much differentiation. There's not resources for differentiation anyway. To have social differentiation, you need extra resources. You need to be producing more than a single person can consume. So there was little differentiation. Also, their food was still mainly collected by hunting and gathering. They have a little bit of domestic cereal. The rest is just collected. They seem to not have domesticated animals yet. It's the very early stage of the Neolithic transition. And the team working there, they believe that these were likely descendants of local foragers. Although the mud brick house culture and some of their tools and some of the ritual elements, they have parallels also in the Levant and in Zagros. These guys seem to, the archaeologists believe that they were somehow local groups who adopted this new culture by cultural interactions with these other groups. But of course, without genetic data, one can not tell for sure whether they were really locals or not. So one thing we're lucky about in the Anatolian Neolithic is that they had a very, pronounced obsession about burying their dead inside the village and even inside the houses, so intramural burial. So this is one of those graves. They were digging up graves inside their houses and burying the dead and covering and then sleeping on the grave, basically. Some of the people were also buried outside the houses. Some of them were buried in the toilet areas, so the patterns are a bit interesting. We still are trying to interpret what these different burials meant, whether those people were treated differently, whether there were different family groups, whatever. Still, we're working on it. But, okay, we get a lot of, the advantage is compared to burying your dead outside, it's very easy to, while the archaeologists are digging the settlement, we get a lot of bones, we get a lot of bones. And the question is, can we get any DNA out of these bones, which are already 10,000 years old, which is pretty hot. Again, people in Leipzig 10 years ago would tell me it's impossible to get DNA out of Turkey. I was actually asking one of the people there, the director, and he was saying about whether it might be possible eventually to do ancient DNA in Turkey, and he was saying it's too hot, so nothing would be preserved. But that was the time when people were still doing Sanger sequencing. And so now that we have this new approach, next generation sequencing, we tried this is our lab in the Motsumer building, not in the biology building, just to keep it more clean. You powderize your sample and you extract DNA and you do shotgun sequencing. So out of about 20 libraries we sequenced, from most of them we didn't get anything useful. From about 3 of them we got about 1% of human DNA. And from one of them we got 6% human DNA, and we were super lucky, super happy with that. And further, my co-PI, who's running the lab, she tried this capture approach. So you can, just like when doing exome capture out of randomly shared DNA, you can enrich for exonic DNA and sequence only that. You can also use a similar approach to capture human DNA out of your library to enrich for the human DNA, and thus you have to sequence less to get the same amount of information. So she used whole genome capture, this is Füsun, and she could increase the endogenous DNA proportion quite a lot, not super dramatically, but at least in our best case it went up to 20%, in some it went up to 5%. And we continued sequencing. That is also thanks to, at the time we didn't have much money, so Swedish people were kindly, generously contributing there. Of course, one also has to check if the data is authentic or not, but we see that here is the length distribution of the molecules we are sequencing, and most of them are around 50 base pairs, as you see. And then this is just one example library. When we check the post-mortem damage signature, we also see that they have accumulated the C to T transitions at the end, so that also makes us happy. And as I mentioned, you can also check other things. For instance, if it is authentic, if the DNA is authentic, then you expect to observe only one mitochondrial haplogroup, because you only receive your mitochondria from the mom. You cannot have multiple moms, right? So you can only have one mitochondrial haplogroup. That is another way to ensure that your DNA is authentic. Okay, and the first question we asked was whether these guys could be migrants coming from, as I mentioned, they had somehow adopted this village life after people in the Levant and in Mesopotamia. So the question was whether they might have they might actually be representing migrants that came from these other areas, or whether they might have adopted this culture just by cultural interaction. And to address this, we joined our data with published, other people have also been publishing ancient genomes from the region. And we already had genomic data from, for instance, hunter-gatherers from Europe around the same period. hunter-gatherers from Caucasus and Iran, and also farmers from Iran, again from the same period, and hunter-gatherers and farmers from the Levant. And this is Boncuklu, and this is another site we had sequenced. So we added our data on top of what we had here. So I should also perhaps briefly explain how this analysis is done. Basically, you are doing pairwise comparisons between different genomes using all information across the genome, and you can then summarize these pairwise differences into an MDS plot as a summary of the genetic distances. So the more distant 2 points are, the more different they are. So these two are more similar to each other, these two are more different from each other. And further, you can do tests, more explicitly a test whether these two guys are more genetically similar to each other than others. So these lines you see, the ellipses indicate that they are indeed more similar to each other than to other groups. And what we see here is that the three regions which encompass this Fertile Crescent area, the Levant, the Zagros, and Central Anatolia, they all had distinct gene pools. So the people living here were genetically distinct, which implies that they had actually developed this new culture, this Neolithic culture, and shared domestic species, not by moving into each other's area, but just by cultural interaction. So we know that, for instance, some species were later, in sheep for instance, they were domesticated in Central Anatolia, and obviously they were transmitted to other regions by cultural interaction. And probably barley was first domesticated here, and it turns out that it was transmitted to Central Anatolia again by cultural transmission. So this suggests that people were kind of nice to each other, and they were open to share and exchange their know-how and also their products. And Gülşah, who'd done her PhD here, she was the person who ran these analyses. There's, of course, you might also ask what happens afterwards. Okay, Neolithic culture had matured here until about 7000 BC. But afterwards, we see in the archaeological record again, we see the expansion of Neolithic villages into West Anatolia first, and the Balkans, and through the Mediterranean to Iberia, and also to the north, Central Europe, Scandinavia eventually, and also goes eastward into Iran and into India eventually. So this is the center, and the culture spreads. And again, a long-standing question. This is a representation of how it spread over time to Europe, and the dates indicate the first evidence for Neolithic settlements in different regions. As you see, it still took quite some time. From 7,000 reaching here, it took about nearly 2,000 years. It wasn't super rapid, but given that, it's still archaeologically, it's still relatively fast process. The question again is, was the adoption of the new Neolithic culture driven by cultural interactions? So did the European hunter-gatherers adopt this new Neolithic culture, farming and living in villages, or was it that people were actually moving in to Iberia or Central Europe or Scandinavia? What do you think? Both could be. There was a lot of debate. So first people thought it must be people moving in. But then some archaeologists were saying, no, it should be indigenous development. And then some other people saying, no. Again, just studying the archaeology, it was not super clear, because there was also changes in the material culture along the way. So it was not really easy to say which model fit. But when you add onto the same plot, when you add the genetic profile of the first European farmers from Iberia, from Scandinavia, from Central Europe, and also Aegean farmers here, they cluster very closely with the Central Anatolian farmers. So whereas the European hunter-gatherers of the same period, they are over here. So the first farmers in Europe were obviously migrants. They actually And interestingly, it also turns out that for some hundreds of years, sometimes thousands of years, they interacted little with the local hunter-gatherers. And any interaction might have been adverse interactions, potentially. So although Europe is today pretty, how to say, not very welcoming to migrants, actually modern-day Europeans are at least 50% migrants. We now know, again, thanks to other work done, we now know that modern-day Europeans' ancestry comes from, in all regions, it comes from three sources. So one is Anatolian farmers, one is European hunter-gatherers, and then there was another wave of migration in the Bronze Age from the Ukrainian steppes. So now they are a composite of basically three gene pools. And if we had added modern-day Europeans here, they would be somewhere here. Okay, that actually brings us to a related topic. So how during this Neolithic transition and afterwards, how diversity changed within local gene pools. And you already, people had already noticed this, that if you compare a European farmer and European hunter-gatherer, the hunter-gatherers in Europe had much lower diversity. Whereas the farmers, these are Scandinavian farmers, they had much higher diversity. And modern-day Europeans, they have even further diversity. And when we added to this data our own Boncuklu farmer, interestingly, so it had the earliest farmers we find in Boncuklu in central Anatolia, they had pretty low diversity, similar to the hunter-gatherers in Europe. So farmers from Anatolia from later periods, they seem to have higher diversity. So how can we explain this result? Of course, it's not mutations accumulating. Mutations don't accumulate so fast. It's mainly people mixing up. So over the Neolithic, when people, it's kind of ironic that people adopt sedentary cultures, so they become more sedentary, but that the consequence is that the birth rates increase. So the more sedentary you are, you can produce more children. The more mobile you are, you're restricted on. So usually hunter-gatherer mothers can have kids every 4 or 5 years. Whereas if you're local, you can have kids every 1-2 years, much faster. So becoming more sedentary and also food production, it allows population growth. And as population increases, people start moving around more often. And then also mixing with other groups. And this is the process we were observing. I think I'm running out of time. I can skip this part. I can then briefly talk about what happened afterwards. So you might wonder, so this is another representation of the previous graph was a summary where each point was representing just the population average. This is basically the same data, but where not populations, but individual genomes are represented, or then again done by Gusha. And you might ask, okay, within this graph, where would modern populations fit in? And it turns out that the modern-day populations who are genetically most similar to Anatolian farmers, the earliest Anatolian farmers, the Neolithic farmers, are Southern Europeans, like Sardinians or Iberians. Whereas the modern-day Anatolia is somewhere here. And how could you explain this? So for one thing, so after this migration event to the Neolithic migrations to across the Mediterranean, these guys, especially people in Corsica, in Sardinia, they remained relatively local and isolated. So they retained their Neolithic farmer genetic profiles. Whereas the farmers in Anatolia, they were subject to incoming gene flow from many regions in the following periods. So that has caused the shift from, of course, some of our ancestry does go back to the Neolithic times, but incoming gene flow from other regions, it has also differentiated the gene pool here. And we see the same also in Iran. So for instance, when you compare Neolithic Iranians with Chalcolithic later coming and modern day Iranians, they have shifted their profile a bit. if you look at Greece, Neolithic Greece, modern Greece, there's also some shift, and this is work done by Dilek in our lab. And if you compare Neolithic Anatolia, Bronze Age Anatolia, and modern day Anatolia is also pretty diverse, as you see. There is a shift, and we see, and that shift, those shifts seem to be contributed both by Caucasian gene flow from the Caucasus, from the east, and also from East Asia, and also a bit back from Europe. So this is actually data published by a Danish group recently, and it's just a presentation of that. Okay, and finally, I will briefly mention what kind of functional changes might have occurred since that time, within the last 10,000 years in Anatolia. We only just recently started looking at into this. We already have, well, these genomes we have published, 9 genomes from Central Anatolia, from Neolithic times, and some other group published, actually 2 groups published 21 genomes from northwestern Turkey, again from Neolithic times. And we are comparing that with 16 present-day, relatively high-quality genomes from different parts of Turkey. And we are, for different loci across the genome, for different functional loci, these are phenotypes we collected from the GWAS catalog. And for instance, for hemoglobin measurement, we have a bunch of SNPs, and we measure the frequencies in the Neolithic Anatolians, in modern day Anatolians, and we calculate differentiation. FSD is a very commonly used statistic for population differentiation. And we do this for functional SNPs. We also do it for neutral, so non-functional loci across the genome. And already, if you study non-functional loci, you find a differentiation statistic which is around 0.4. And you can then ask, do we find any functional groups which have differentiated more than non-functional loci? And that could potentially indicate positive selection driving allele frequency change. And if you run this analysis, well, with our limited data yet, there's nothing that passes multiple testing correction, but it's noticeable that one of the strongest signals was coming from interleukin measurement. So things related to immunity, some processes related to immunity seem to have changed their frequency more often than neutral loci. But again, this is something we are still working on, and it's not, I'm not saying it's a significant result yet, but with more data, potentially we might find some real signals. You might also notice that many groups actually have less differentiation than non-functional loci, and that's the outcome of negative selection. So if allele frequency change, if the phenotype is somehow optimized by selection, then any allele frequency change would also be selected against. So we would find less differentiation between past and present in such loci. Okay, I think I can finish there. So a lot of people, of course, contributed to this, both working, well, let's start with the archaeologists, people working in different settlements in Turkey, Neolithic settlements, and also the anthropologists working with them. People working in the wet lab, especially I should thank Füsun, who's a very dedicated person, and who soon I hope she will be professor at one of the universities, Ankara. Gülşah led most of the dry lab analysis, so in silico work on the Neolithic genomes. And this guy, he's in Stockholm University. He generously was funding us when we didn't have, at the time, when we didn't have much money. Okay, yeah, I didn't add I should also have added ERC there. We are also using ERC money for the last 6 months now. Okay, thank you.
We thank Mehmet for this great talk. So the talk is open for questions from the audience. Yeah.
So I'm really not very familiar with what happens in this Neolithic period.
Uh-huh.
How about the exchange, or what is it called, buying things from each other? When does that come
I guess there was some type of barter. Of course, there's no money. Money is super recent invention. And also trade, as we know it, is probably, I mean, there's no surplus during most of this period. I mean, the type of surplus and interactions and property as we know it, it mainly is established much more recently in the Bronze Age, like 5,000 years ago or something like that. But we know that they're interacting because, for instance, we know that obsidian, so those black stones, this is volcanic stone, can be very sharp and super useful. It has sources in Central Anatolia, and Central Anatolian obsidian was traveling all the way to the Levant. We find the Central Anatolian obsidian tools back in Levant, down in Levant, I should say. And we also find some Levantine, for instance, shells coming from the Mediterranean coast up in Central Anatolia. So there was, we already know, already before the Neolithic, hunter-gatherer groups had these exchange networks. So exchange was not something totally new to them. How they decided to exchange what, this we don't, we're still trying to understand. And what we now wish to do is, working together with archaeologists, to sequence many genomes, as like from 20, 30 settlements across Anatolia and the region, and to make a map of gene flow across settlements, and add onto this a map of archaeological material culture exchange. So exchange of ritual elements, of raw material, and also sheep, this kind of stuff. And then try to see what items actually were following gene flow and what items did not follow gene flow at all. So to be able to interpret what archaeologists are saying more. But yeah, it needs, of course, a lot of effort from the archaeological side as well. There are no such comprehensive data sets yet also in archaeology. Yeah.
Thank you for the follow-up. I want to ask you that we saw that from the genetic information that Anatolian people, Anatolian Neolithic people, migrated to Europe, right?
Uh-huh.
So, but
yeah.
We don't see pre-Neolithic people. So can we say that these people killed those people?
No, okay. Yeah, it's good point. So in this graph, so these are hunter-gatherers from mainland Europe and Eurasian steppe. And these are the European farmers who are genetically very similar to Anatolian farmers. What happened was, initially there were two groups living in Europe, some doing farming and the other ones trying to continue with their hunter-gatherer culture. And eventually they mix. It takes some time, so sometimes 500 years, sometimes 1,000 years. Again, the data is kind of sparse, so we cannot tell so well, but it does appear that it takes some time. We do find sometimes some hybrid individuals, like individual hunter-gatherer background, genetic background in a Neolithic set, buried in a Neolithic setting. So there are a few cases like that. But overall, till we find homogeneous populations that are a mixture between these two, who would stand here, it takes some time. So they don't disappear, but they just mix. How they get assimilated, how they adopt farming culture, this again is something the archaeologists have to tell us. But yeah, we know that they did contribute to modern day gene pool. They didn't disappear. But most likely the first interactions might have been a bit not so nice, because the farmers were probably opening up a forest area for farmland, and these guys were hunting or collecting stuff in that area, so there could be some conflict of interest, but we don't know. I'm
curious, is it possible to extend a similar analysis to ancient Greek civilization and correlate your results with what is written records, for instance, and the histories of Herodotus is talking about certain Greek tribes
moving in, yeah.
Actual names then, and with the time being closer today, maybe you can have a higher resolution.
It's very true, and we're interested in that as well. So we're collecting data from more recent periods as well. So for instance, we know that there was a Celtic expansion. The Celts were actually a tribe living in Central Europe, and then some of them went into Western Europe, and the Irish languages, Irish and Scottish, are remnants of that expansion. And the East, they also expanded east, and this was the easternmost border. Actually, Ankara was part of Galatia, so the Celtic region of Rome. And we have some, for instance, burials with Celtic culture. And we are also, for instance, now running analyses on this. But I have to tell, the closer you get in time to today, it will become a bit more difficult to see differences, because the populations become more mixed up and more homogeneous. So we will need larger sample sizes to tell apart small events. Here, because by the early Holocene, so at the end of the Ice Age, humans living in Eurasia and also other places, they were usually small, relatively isolated groups, and isolation means genetic differentiation over time through genetic drift. So it is easy to see differences. So when some group moves over to some other place, they come with their distinct genetic profile. Whereas when you move forward in time, those distinct profiles become mixed up and more vague. But yeah, we'll be doing all our best. We're also collaborating. We're trying to collaborate with Greek and Iranian and Armenian archaeologists, anthropologists, ancient DNA groups, because the US and Europe, they are behaving super imperialistly with respect to ancient DNA. They're collecting samples like crazy everywhere to, pick up all the low-hanging fruit in archaeology and history. And answer, because it's, this analysis is not super sophisticated. So as long as you have money to do sequencing, you can prepare libraries and you can sequence. You can answer any obvious question, and you just need the material. Material is the limiting factor, and they send scouts over to archaeological excavations, and they promise a nature paper, science paper, give us your samples, and it's frustrating us a lot, unfortunately. Yeah, we are very, how to say, sometimes we really get pissed off at these groups. What I wish is would be very nice if everybody set up ancient DNA labs in their own regions and were collaborating, producing data together, analyzing data together, sharing data. Not all science has to be done by rich countries, I guess.
Is there like a protocol to move bones from like the east to your lab? Is there like a special?
Yeah, you need to have so in every country has their own regulations, but in Turkey the regulation is that the archaeologist has to say yes, and the museum that the archaeological excavation belongs to also has to say yes. So it's pretty well regulated. And also exporting samples, it also needs extra permission from the museum. And luckily for us, some years ago, there was a rule added, a regulation that if an analysis could be done in Turkey, it should not be, the material should not be exported. So this is, of course, a lot to our benefit, but still, people find their ways ways around these rules, and a lot of interesting stuff. Other groups with more money, others. Of course, we're also a bit uncomfortable we're currently the only group doing this type of ancient genomic analysis. We wish there would also be more labs in Turkey, so we would not appear as a monopoly here. We don't want to be, restrict the research, and also our capacity is limited, so we cannot process more than 500, 1,000 bones per year. So, and there are a lot of groups interested in this.
Okay, so I know that the high school history classes were really boring, but it's more fun in this way. So thanks again, Mehmet, for being here.
Thanks again for. Merhaba.
Daha önce tanışmıştık, mailleşmiştik. ERC sunumunu paylaşmıştınız benimle. Hatırladınız mı?
Evet. Nasıl oldu?
Valla olmadı.
Denemede.
Hayır, bu ikinci defa gösterebileceğim. Yine olmuyor.
Görüşürüz.
Sana yazacağım o.
Tamam.
Olivier galiba
Ha, evet.
çocuğun adı. Bilkent'ten. Siz Bilkent'ten misiniz? Hangi bölüm? Arkeoloji?
Hayır, fizik.
Ha fizik tamam. Okey. Bulursam o kişiyi yazacağım
Siz
sana.
sağ olun.
Hadi görüşürüz.
Konuşmak için. Teşekkürler. Sizlerle yine görüşmek üzere. Nasıl gidiyor?
Daha önce konuşmuşluğumuz var da bir teşekkür ederim.
Yok, estağfurullah. Ben konsolideydim mi isterdim? Görüşürüz.
Start bitti. Şimdi artık kontrol ediyoruz.
İyi puan aldınız mı? Almışsınız
Yani
zaten.
puan bizde bir puan değil mi? A diyor, B diyor yani. İşte
Tamam.
puan vermiyor. Yani büyük süre çağırdığı zaman zaten herhalde
Şeyin üzerinde B5'inde oluyor.
Evet. Ama mesela bu ikinci gidişimde, ilk gidişimde 11 erken bakmıştı. Bu gidişimde 9 erken baktı ve bunların çoğu işte bir device yapacağım ben. Device parmaklarını getirip söylememişsin diyor. Ama daha önce bakan 11 hakemin bir tanesi bile böyle bir şey söylememişti mesela. Yani belli ki bunlar bir tanesi ortaya bir şey atmış, diğerleri de üstüne atlamışlar gibi geldi bana. Öyle yani, sinir oldu. Yapacak bir şey yok.
Yani
Hatta aklıma şey bile geldi.